All right, I want to start with being a little transparent about your grade that I entered into Blackboard. Next week, hopefully next week at some time, I will show you the 5 most commonly missed questions. I can't do that yet today. There were a few students with extenuating circumstances that haven't taken the test yet. But I can share kind of in general the statistics here. The curve, the grade that was posted on Blackboard reflected a little bit of a creative curve. I took the total number of questions, 62, and multiplied every wrong answer by 0.9 instead of 1. So everyone you missed was minus 0.9. And then I added on a point. Okay, so with that formula, that gave everybody a little bump up, kind of depending how many you missed. But with that curve, the 2 people that only missed 1 got 100%, and then on down from there. So we had about 10% of the class end up with A's, which is a pretty reasonable number. A little higher on the other end than, of course, we want to see. But not a bad first test. Without the curve, the mean was somewhere around 70, 71%. With the curve, the mean was 74%. The people that took it in class actually did much better on average than the people that took it by respondus. So I'm not quite sure how to explain that, whether the respondus people have been watching all from home and that's just not as engaging. So, you know, however you are listening to the lecture, whether you're here or whether you're at home, you might want to think about maybe it's better to come more often. Give that a try. I know that there are circumstances that are keeping people at home. I do want to have as many of you as possible take the exam in person next time. So I was kind of lenient about that. Some of you just, it was more convenient to stay home. I really, due to some issues with respondus and just keeping the integrity of the exam, I do want to have you come in person if at all possible next time. So we'll talk more about that. If you do want to come and see your exam, I've had a few people do that already. Of course, it's hard. I can't have a mob of people in my office at once, but you know, make an arrangement to come see me. I'm around a lot and take a look at that. Or if you have any questions like, oh, this one question is really bothering me, and you know, we might be able to do a little bit by email or Zoom too. But again, I will post some of the, we'll talk about the more commonly missed questions. The ones that were missed the most, there was a couple that only 30% of you got right, but there wasn't any one question that like everybody missed. The people that only missed a few, I went through and looked at what they missed and made sure I didn't make errors on answer keys. And so, okay. All right, so moving forward here, we started last lecture just kind of a review of key concepts of metabolism. We're going to finish that up today by talking a little bit about enzymes before we're going to hit the pathways. So I'm going to throw a lot at you today. Hopefully you have a little bit of background in metabolism, but we are really going to talk about things fairly generally. So there's not going to be memorization of pathways or structures or enzymes or any of that. So hopefully it's, again, very, very doable, okay? All right, so this should be a review. You should know a little something about enzymes. Of course, last time we talked about redox reactions and electron carriers and ATP. It bears repeating. Enzymes, of course, they are typically going to end in ASE. Now, that's not true for all enzymes. Trypsin, for example, is an enzyme. It doesn't end in ASE. But if you see a compound that ends in ASE, it is an enzyme, okay? If it ends in OSE, then it's a sugar. But ASE, it's an enzyme. Of course, what do? They lower the activation energy of reactions. So for example, most of the reactions that take place in our body would take place without an enzyme, but they would happen so slowly that we couldn't sustain life. So here, you know, the energy it would take without the enzyme, and here with the enzyme for the reaction to happen. Okay, let's see. So they speed up reactions. Little terminology. Enzymes work on substrates. Usually we write the enzyme over the arrow like that, and what we get are some products. Okay, here they said reactants instead of substrates. That's the same thing. Okay, so the enzyme, of course, is not changed or used up in the reaction. The substrate, however, is changed into the product. So if we want to see if a cell is able to make a particular enzyme, typically what we'll do is look to see if certain products show up. And you'll be doing that in lab next week. Sometimes it's more appropriate to see if the substrate disappears. Sometimes that's a little bit easier. But of course, there are some enzymes that are common to almost all life, but then we get, especially certain bacteria can make certain enzymes, other organisms can't. And that's one way we tell them apart. Let's see, if the enzyme is only composed of a protein, we call that a simple enzyme. Okay, but a lot of enzymes actually are not just protein, they have other parts, the protein part and a non-protein component. Okay, we would call that a conjugated enzyme. So what kind of non-protein parts might be necessary for an enzyme to function? If you had some kind of inorganic ion, we would call that part a cofactor. If we had some kind of organic non-protein components, then we would call that a coenzyme. All right, so the next slide kind of, the next graphic here illustrates that a little bit better. So if you have a protein, again, that has non-protein components, the protein part is referred to as the apoenzyme. That's the big blue part there. If you have an inorganic cofactor, and usually this is some kind of ion, maybe it's iron, maybe it's copper or something like that, that may be necessary to get the active site just the right formation. Of course, the active site, this is where the substrate binds. There might be an organic cofactor, again, that's necessary for the right shape there. And all of that together is the holoenzyme. All right, so talking, so I didn't post this. This is a graphic from your book here, but just illustrates, you know, how one enzyme works. It's one of the enzymes in the pathway of glycolysis. Okay, don't memorize the specific enzyme, but here's the enzyme. That's the blue part here, and here's the substrate, fructose 1,6-bisphosphate. The enzyme binds to the substrate. We have an enzyme substrate complex. In this case, what that enzyme does, this is the splitting step in glycolysis, where it splits that fructose 1,6-bisphosphate. And here's the products that we get from that. We get two different products. And then the enzyme just recycles back and binds with the new product. Okay, so just an example to look at what kind of things are going to influence enzyme activity. You should recognize these as the same sorts of things that influence the growth, the rate of growth of bacterial cells. So kind of the first thing that we always think about is temperature. An enzyme typically has some ideal temperature where it's going to work the quickest. That usually matches the ideal growth temperature of an organism. When you get a little higher than that, it very quickly becomes either inactivated or denatured. pH, I'm not sure if they're still doing that lab in Bio 120 with the catalase, where you'd put the paper discs in the catalase and then see how quickly it rises up in the water column, but certainly enzymes have an ideal pH where they're going to function the quickest. Activity falls off when you get too far one way or the other, and typically that's going to be around neutral, but be different for different enzymes. And if you alter the substrate concentration, so back to our example here, if there's very little of this substrate, then you're not going to get very much product. That reaction just is not favorable to happen. So the more substrate that you get, the more enzyme activity, but there is some saturation point. So as soon as there's, at some point there's few enough enzymes that it can't keep up. Same thing if you altered the enzyme concentration. The more enzyme you get, as long as there's plenty of substrate, you're going to get a faster rate of reaction. But again, at some point that will kind of level off. All right, so typically a cell, we're going to talk more about regulation in a few weeks when we talk about in our genetics unit, but typically a cell is not going to make an enzyme unless it really needs it, if possible, because energetically it's pretty expensive to make any protein. But the cell does have some ways, once an enzyme is made, of turning it on or off. So if it's necessary or if it's not necessary. So we have a couple different kinds of inhibition. Competitive inhibition, this is where the inhibitor binds to the active site. Of the enzyme. So in this case, it's going to be a competitive hit inhibitor. It's going to be a molecule that really mimics that part of the substrate that would fit here into the site. Okay, and it fits in there instead so that the substrate can't bind, and then you don't get any product. Okay, fairly straightforward. You could have an inhibitor that doesn't bind at the active site, and we call that a non-competitive inhibitor. Yeah, question? Yeah. Whoops, it's not going here. Oh, there it is. Sorry. Sometimes it takes a second to catch up to me. Okay. All right, you can't have an inhibitor that doesn't bind to the active site. We call that a non-competitive inhibitor, or also another name for it is an allosteric inhibitor. So allosteric, that just means other. So some site other than the active site. So in this case, here's an allosteric inhibitor that binds, say, maybe at this side of the enzyme. Of course, this is the active site right here. But that binding of something over here actually distorts the active site a little bit so that now the substrate doesn't bind anymore. Maybe a better name for this is allosteric control, because you can also have an allosteric, some kind of compound that binds allosterically that is necessary for the reaction to take place. So maybe you have some compound that binds over here that now changes the shape of the active site in order that the substrate only now fits. Okay, so again, better word maybe is allosteric control. I'll write that up here. And an example when that happens is where we see that allosteric control is during feedback inhibition. So if you have some pathway where you're getting some end product here. So this happens a lot in metabolism or biosynthetic reactions as well. So here you have an enzyme here that binds to some substrate. Okay, you get one product. Oops, so that's the enzyme substrate complex, and then you get some intermediate. You have another enzyme in the pathway. You get another intermediate, maybe a third enzyme, and you end up with the end product. If a cell is having a bunch of this end product build up, if it's getting more than what it needs, it makes sense for that end product to actually perhaps bind to that first enzyme in order to keep that reaction from happening anymore. Pathway shuts down. Okay, so it makes sense. You get too much of something build up, you have a mechanism so that you're not getting more and more of it if that's not what the cell needs. Yeah. So that's a good question. Can this be competitive and non-competitive for feedback inhibition? I believe so. Yeah, as far as I know, you'd have to look at a particular pathway. This is sort of a general scheme, but I can envision that that could be the case. Okay. All right, so as we move forward into our pathways, we're going to start with glycolysis. We're going to try to keep in mind some kind of big picture things. So as we look at these pathways, we're going to look at how much ATP does the cell have to spend to start it up? How much does it harvest from that pathway? Because that's really the primary thing that we're interested in here. Metabolism, that's the process of a cell gaining energy. That's the whole point in order so that it can grow. We're not going to memorize pathways, like I said. We're going to look at key juncture points, and we're going to think about kind of what we start with and what we end with. But again, we're not worried about all the intermediates. So again, thinking about what we end up, what the cell gains from using that pathway. I will be pointing out a few things about the pathways themselves, just mostly for context. It's always helpful to follow the carbon. So if you're starting with 6-carbon glucose, it makes sense that we're still, that carbon just doesn't disappear, right? We end up with 2,3-carbon pyruvic acid. Okay, so we'll that helps us kind of keep track of things, thinking about the carbon. And really importantly is what happens to those electrons that get peeled off as part of redox reactions, because those electrons are going to be converted into lots and lots of energy at electron transport chains. So really important to pay attention to where those electrons are going. Okay, so this is your book's kind of graphic overview of metabolism. And I'm going to come back to this fairly often, actually, because it's, again, we're kind of thinking big picture type stuff. So first of all, what are we starting with? Usually glucose. So whenever you talk about metabolism, you're almost always talking about some kind of sugar being broken down, because that's the most readily available energy source typically in a cell. That's typically what the cell is going to choose to use first. We'll talk about what happens if only proteins are available or only lipids are available. But in general, we think about what happens. Again, that's the first choice of a cell to use for energy. Okay, notice the green side here, respiration. We're going to start with that, thinking about respiration. And we're also then, I don't know if we'll get to it today, talk about fermentation. So basically, respiration involves glycolysis. We'll transition step here with acetyl-CoA, Krebs cycle, and an electron transport chain. It doesn't have to involve oxygen. Okay, for us, we always need a final electron acceptor, oxygen, but there are many, many bacteria that can use something else besides oxygen. So if they use something like nitrate, okay, that's still respiration, but we call it anaerobic respiration. And it's still the same basic pathway. There's going to be some differences in the Krebs cycle that we don't really worry about, but that's still the same, again, basic pathway involving Krebs cycle, electron transport chain. Okay, fermentation doesn't have an electron transport chain. It's much simpler. People tend to kind of group anaerobic respiration and fermentation together because neither one of them require oxygen, but they're really very different processes. And sometimes we group them together because we can't really tell, you know, from whatever test what the cell's actually doing. But we're going to talk about respiration first, where that takes place. There are some alternatives that some bacteria use to glycolysis. In general, though, glycolysis is an extremely conserved process, meaning almost all of life does glycolysis. There's a few bacteria that aren't able to, and we'll talk about that, but extremely, extremely common. Okay. All right, so let's focus on glycolysis. I do like to use the graphics in your book, but sometimes I will pull one from somewhere else that seems to show something a little bit better or a little bit more clearly. First thing I want to mention, in metabolism, we often have different names describing the same thing. And that's really not to confuse you, but we often will have a name for a pathway, for example, that's really good and descriptive, like glycolysis, the splitting of sugar. It's a great name. It describes exactly what's going on. We often have another name for a process that gives credit to the people that elucidated it. So I asked this as part of a multiple choice clicker question last semester, and I think only 10% of the class could even recognize it in multiple choice. But the other name for glycolysis is the Emden-Meyerhof-Parnas pathway. Or sometimes it's just called the Emden-Meyerhof pathway. All right, you really should recognize that. Sometimes you'll read in a research paper them talking about Emden-Meyerhof pathway. You should know, oh, they just mean like glycolysis. All right, so same thing. Think about where this is taking place in the cytoplasm of cells. Both prokaryotes and eukaryotes, this takes place in the cytoplasm. All right, so I like this picture because, again, I think it's great to focus on structures and enzymes, but you really kind of appreciate what's going on and see the elegance when you see what's actually happening, but we just don't have time to do that in here. We'll save that for your biochemistry classes. Sort of in general, what's going on here. That's why I like this graphic. So you're starting out, obviously, with glucose here. You have to input a little bit of ATP to kind of get the process rolling. So same thing in life. You usually have to spend some energy to do anything worthwhile. All right, so invest some energy. And then this is the splitting stage right here. The fructose 1,6-bisphosphate is lysed. This is not a symmetric molecule, so the splitting part, you don't get two equal things from that. But the one molecule usually is converted right into the other. This graphic doesn't actually show that, just for simplicity. it just says, well, you get 2 molecules of this 3-carbon glyceraldehyde 3-phosphate. So cleavage step there. And then the bottom part of glycolysis, you're getting some electrons in the form of the electron carrier, NADH, and 2 ATPs here for each glyceraldehyde 3-phosphate, and you end up with 2 molecules of pyruvate. Same thing as pyruvic acid. Okay, so, all right, so thinking about our net gains, here we get 2 ATP. Again, you put in 2 to begin with, but you get 4 out at the end. You get 2 NADHs. Those electrons, again, are going to be used, the electron transport chain, in order to be converted into ATP later, and 2 pyruvate. Okay, so really not a lot of energy, but some. Okay, I didn't post this, but I wanted to make you aware, if you actually have a 5th edition textbook, the graphic that they posted for glycolysis, in the 4th edition, they just basically said the splitting step, they said, oh, well, now you get 2 of these and 2 of these, and they decided, well, that's not very clear, we should show it splitting. So they did that, but then they still kept the 2. So you end up, you know, this says that you end up with 4 pyruvic acids. Of course, that doesn't really make sense. So this should just be, you know, 1 of each. Don't let that throw you if you're using an old edition. They fixed that in the 6th edition. Okay, so let's focus here on again, here's the Emden-Meyerhof glycolysis, just shown a little bit different way than the previous example. Again, I don't need you to memorize all these intermediates. Maybe you have them memorized from biochem. I don't, that kind of thing doesn't really stick in my head very well. We talked about our net gains here. I wanted to show that up here. This graphic does show the dihydroxyacetone phosphate that typically gets converted into glyceraldehyde 3-phosphate. I wanted to put that up next to a different pathway called the Entner-Doderoff. Kind of sounds like Emden-Meyerhof, but this is a pathway we're going to call an alternative to glycolysis. Only some bacteria can do. Remember I said almost all of life does glycolysis, but the Entner-Doderoff, there are only some bacteria can do that. There are bacteria that are missing, I think it's this enzyme right here, it's phosphofructokinase. They're missing that enzyme, so they can't do glycolysis. We now know that actually that Entner-Doderoff pathway is a little bit more common than what we once thought. There are some bacteria that will do this pathway instead of glycolysis when you grow them on certain medias, or when they're growing in certain parts of the body, for example. So it's a little bit more common, but here's just an example of some of the organisms that can do that. I just threw that up there because typically we use Pseudomonas a lot in the lab. We use Enterococcus in the lab, and of course E. coli. Those are organisms that you may recognize, but again, don't memorize that. So what's the difference between the 2 pathways? Like I said, it's the up top part here that's a little bit different because of the absence in some of these organisms of this enzyme down here. Right away at the top here, you'll notice a difference. You have to input some ATP, but then we get a reduction of the electron carrier NADP. Okay, so in glycolysis, we only had NAD. NADP is NAD plus a phosphate. That phosphate tags those electrons to be used in biosynthetic pathways. Whoops. Okay, so those electrons carried by NADPH don't go to the electron transport chain. They go to be used in some anabolic biosynthetic pathway. Okay, and then we get a little different intermediate here. Actually have it written what that stands for, but I don't have it memorized. And then right away, we get 1-pyruvate produced. Now, you get that pyruvate without getting any of the energy or electrons out of it, basically. But you still end up with a pyruvate. This KD PA gets split into, again, 2 3-carbon compounds with the glyceraldehyde 3-phosphate. Now, this is just the same as over here in glycolysis. So we get out an ATP, 2 ATPs, and NADH. Okay, so if we think about the net gains of the Entner-Doderoff, okay, we're only getting half the number of ATP. We're only getting 1. We input 1, we get out 2. We're getting 1 NADH and 1 NADPH. But we still got 2 pyruvate. Okay, so for organisms that are relying on glycolysis for energy, this is not a good solution. So typically organisms are aerobes. They're actually getting energy from all of these electrons at the electron transport chain. And so the fact that you only get 1 ATP instead of 2 is not a huge problem here. Okay, so organisms typically only do one or the other at one time, certainly. They're not going to be doing both of these pathways. Okay, there is a third alternative to glycolysis called the pentose phosphate pathway. And by the way, your book talks very little about Entner-Doderoff, which is kind of surprising because it's something that just bacteria do, and it seems like we should be focusing on. It talks very little about pentose phosphate pathway as well. I think it's maybe in the index, the pathway. But again, this is another alternative to glycolysis. We can do, humans do a pentose phosphate pathway, bacteria do it, and they typically are going to do this when cells' needs are biosynthetic. So in other words, they don't need energy, they need precursor molecules in order to make cellular components. Okay, so a cell would maybe be doing both glycolysis and pentose phosphate pathway at the same time, or maybe more of one than the other. Okay. All right, so this is a very complex looking pathway. I want to pick it apart and kind of tell you kind of what I think is more important. This does have some other names. I will always call it the pentose phosphate pathway. Hexose monophosphate shunt is another name that you might see, but I always call it this. So we're starting out with glucose again, glucose 6-phosphate. That's the same as glycolysis, Entner-Doderoff. But then right away here, you see a difference from glycolysis. We're getting a reducing power, again, in the form of NADPH. So remember, those go towards biosynthetic reactions. just like they did in the Entner-Doudoroff. Okay, so here, 2 anabolic reactions to power those and make them more energetically favorable. And then there's lots of branch points in this pathway, and it all depends on what the cell needs. We could end up with ribose 5-phosphate if the cell needs to synthesize nucleotides. Okay, so let me write this clearly here. All right, so key pentose sugars can be produced. Okay, we'll talk about the structure of DNA probably next time and RNA, but remember they have 5 carbon sugars as part of a nucleic acid. So in order to synthesize nucleotides, this pathway can give us precursors there. If that's not necessary, it can follow a different pathway. This erythrose 4-phosphate is used to synthesize amino acids. Okay, so I'm just going to say a key precursor for the synthesis of amino acids. And those actually, in bacteria, this erythrose 4-phosphate is necessary to synthesize aromatic amino acids. That means amino acids that have a ring structure in them. Humans can't do that, but some bacteria can. Okay. And you can see here, I kind of skipped over this. Here's glyceraldehyde 3-phosphate. That's one of the intermediates in glycolysis. So if, you know, it starts going through the pathway, oh, we need some energy after all, shunts it over to glycolysis, the enzymes of glycolysis. And that's could be what you end up anyway, okay? So again, you're not getting all of these things out of it. It depends on what the cells need, what you kind of end up with. I would say no ATP produced directly from this pathway. Okay, now if the cell ends up, again, shunting the glyceraldehyde 3-phosphate into glycolysis, then yeah, it's going to end up getting some ATP out of glycolysis, but none produced directly from this, no reducing power that's going to end up as ATP. Okay. All right, so that's kind of glycolysis and alternatives to glycolysis. I wanted to give you a practice exam question, all right? So I just stuck this in here a couple days ago, so it's not posted. So let's see if you were paying attention about the Entner-Doudoroff pathway, okay? Which of these is true? Is this another name for glycolysis? Is it common to most forms of life? Does it produce half the ATP as glycolysis? It's an anabolic pathway, or it's carried out at the same time as glycolysis. Okay, so hopefully, nope, it's not another name for glycolysis. It's similar, but it's a different pathway. And I talked about there's only certain bacteria that do this pathway. It's not, usually a bacteria will do either Entner-Doudoroff or glycolysis. It doesn't do them both at the same time. And it is a catabolic pathway. Remember, anabolic is biosynthetic, making larger molecules from smaller ones. All right, so the correct answer here is it produces half ATP as glycolysis. Yeah. Okay, so the question is, do we consider pentose phosphate anabolic because we're getting larger molecules, or is it catabolic because it's breaking glucose down? I think the answer to that is both. We call that amphibolic. It's a great question. So parts of it, right, we're building molecules, and parts of it, we're breaking it down. So yeah, that's a great question.
And you said that the pentose phosphate that can be carried out simultaneously or no?
That's my understanding, that yes, pentose phosphate can go on as the same time as glycolysis, or if it's a bacteria that does entner-Doderoff, it can do entner-Doderoff and pentose phosphate kind of at the same time. So we learn about these things kind of by themselves, and then it's really hard to kind of wrap your mind around what really goes on in the cell. And you know, there's catabolic and anabolic and all sorts of things going on at the same time. But we can do the best we can. All right, so continuing in respiration. So we talked about this glycolysis step up here. There's a transition step right here that has to happen with those pyruvates, because that 3-carbon pyruvate can't enter into the Krebs cycle. Sorry, I'm getting a little feedback here when I stand on this side. The Krebs cycle only accepts 2-carbon compounds. So there has to be a little bit of a transition step here. So we take that 3-carbon pyruvic acid, and again, this is still happening in the cytoplasm. We lose a carbon here, so we call that a decarboxylation. Just lose it as carbon dioxide. We get 2-carbon acetate. That combines with coenzyme A. We get a little reducing power, NADH, as a result of that. And then we end up with acetyl coenzyme A. Okay? And then that is ready to be shunted into the Krebs cycle. Okay, so I guess from this little transition step, we could say we gain 1 NADH. Now that's per pyruvate. So if we were saying, you always have to pay attention if I'm asking you, well, how do you count that? Are we asking you per pyruvate, or am I asking you per glucose? We'd be getting 2 NADHs per glucose because there's 2 pyruvates. And then, of course, 1 acetyl-CoA per pyruvate, or 2 per glucose. Okay, so we've got our 2-carbon compound, and let's see what happens at the Krebs cycle. Oh, yep, I'll go back a second. Yep. All right, so hopefully this is the second set of notes that I posted. We're switching into now. Right. So this is a figure from your book of the Krebs cycle. Let's talk about different names for the Krebs cycle. Hans Krebs, he's the one that's elucidated this like mid-1900s. He was born in 1900, so mid-1900s. So I often call it the Krebs cycle. The other names for this, citric acid cycle, because you'll notice here this is kind of the first intermediate we come to, citric acid. That acetyl-CoA binds with the 4-carbon oxaloacetic acid to make 6-carbon citric acid. The other name that you'll see is the tricarboxylic acid cycle, or the TCA cycle. Okay, so those are all the same thing. Tricarboxylic acid cycle comes from the fact that citric acid and isocitric acid have 3 carboxyl groups there. So that's, you know, all 3 of those names, I probably see them all pretty equally. All right. So we start out with this 6-carbon citric acid there once the acetyl-CoA binds to that. And then first thing I want to point out is that we're losing a carbon dioxide here, and we're losing another carbon dioxide here. So we started, we started with 6-carbon glucose. If you take the 3-carbon pyruvic acid, we lost a carbon in the transition step, and here we lost 2 more. So basically, all those carbons that we started with are gone, which is kind of interesting. Some organisms can't afford to lose all that carbon. They need it to build structures in their cells. So some organisms use a modified Krebs cycle where they don't lose as much carbon. So it's called it's some kind of shunt, I forget, but we're not gonna worry about that. But that is sort of interesting. What else are we gaining here? We've got NADH here, and up here, as well as some FADH2. So we're getting lots and lots of electrons, form of reducing power, peeled off of this. Let me start writing this down. So net gains, let's say per pyruvate. Can't spell. Okay. And let's say including the transition step. So again, you always have to pay attention to how you're counting it. So in terms of NADHs, we're getting 1, 2, 3 here, plus 1 other one from that transition step. So 4 NADH and 1 FADH2. Again, those electrons get converted into lots of ATP. What about ATP? We are only getting 1 ATP, again, per pyruvate. And that's actually, the GDP is phosphorylated to GTP, and that's readily converted into ATP. Okay. And we're getting 3 CO2. Not sure if that's, again, not really helpful to the cell in any way. That just gets sort of released. All right. So again, you could count that per glucose. Let's say per glucose, and of course, everything would be doubled. So 8 NADH, 2 FAD, and 6 CO2. Okay, so it's really all about the electrons. So again, I'm borrowing that from that crash course chemistry YouTube video that I put the link up to when we were talking about redox reactions. It's all going to be about those electrons. But I want to stop and give you another practice questions because I thought I would probably forget to talk about this. Okay, how is that ATP generated in the Krebs cycle? We talked about the different kinds of phosphorylation. Remember, that's basically the change from ADP gets phosphorylated to ATP. Is that oxidative? Is that photophosphorylation, substrate level, or a combination of 2 different kinds? Okay, well, hopefully you can cross this out, photophosphorylation, because we talked about that only happens in photosynthesis. And we're not really worrying about photosynthesis here, and because again, organisms that cause disease are not photosynthetic, so we're going to skip all the photosynthesis stuff in this unit. Oxidative phosphorylation, this happens only at the electron transport chain. We'll talk a little bit shortly about why we call it that. What's going on here in the Krebs cycle is substrate level phosphorylation. So the ATP that was gained in glycolysis and the Krebs cycle is considered substrate level. And I was, you know, I was talking to my biochemist husband about this last night. Is this sort of conversations we have at home. And I said, well, it seems like a crazy name because if you look at this substrate here, there's no phosphate there. So where is it getting the phosphate from here? And it turns out this is really just a simplification. that there is a phosphate that does kind of transiently bind. You know, it kind of comes from the medium, basically, or, you know, from inside of the cell, and it does bound there, and it, you know, transfers to the ADP. So it's not a great name that's really describing what's going on there, but that's sort of what we have to live with, I guess. Okay. It's a little bit more easy to see during glycolysis, you know, where the ADP is grabbing a phosphate off of, you know, one of the precursor molecules there, one of the other intermediates. Okay, so we are ready to jump into the electron transport chain. So I just wanted to get our bearings again. So we've talked about all of this that has come off of the Krebs cycle. These electrons now are getting dropped off at the electron transport chain. So this is really where it all comes down in terms of making energy. So your book shows electron transport chain in a couple different ways, and certain ways show certain things a little bit better, so we'll show you both ways. So where is the electron transport chain? This is going to be in the plasma membrane. Oops. Don't give myself enough room there. Okay, so it's very important in both prokaryotes. This is going to be in just the cell membrane. In eukaryotes, it's going to be in the inner mitochondrial membrane. So it's very important that that membrane is intact. Here's outside of the cell, and here is inside. It's not immediately obvious, but I know this is the outside because it's pumping these protons outside of the cell. And those are going to actually convert into energy in one spot where they're allowed to diffuse back in down their concentration gradient. If those were allowed to just diffuse back in wherever they wanted, then we wouldn't get the generation of ATP. So that membrane has to be intact, selectively permeable. And the reason that it's drawn kind of diagonally like this is to try to symbolize that there's lots of potential energy up here at the top, like a ball rolling down a hill. Okay, so once it gets started, all these reactions are energetically favorable until we end up here with some final electron acceptor. Again, for aerobic bacteria, that's going to be oxygen. For organisms that can respire anaerobically, it could be usually an inorganic ion like nitrate, something like that. Okay, what's happening at the first step here, the NADH gets oxidized back to NAD. So that's where the name oxidative phosphorylation comes from, because NADH oxidized back to NAD. All right, so oxygen is not necessarily involved. Even in anaerobic respiration, you get oxidative phosphorylation. Okay, so those electrons get dropped off, and then you see all these electron carriers. They're alternating between their reduced and oxidized state, passing electrons down, and some of them are pumping the protons outside of the cell. So I don't necessarily want you to memorize all those electron carriers, but you should sort of recognize their names. Sometimes they're in a different order in certain cells. Sometimes cells have some of those and not other ones. There's a lot of variability there. So don't memorize that. Some of them are called flavoproteins. They contain flavin, which is a coenzyme. So this FMN, flavomononucleotide. There are cytochromes, proteins that have iron that helps them, again, convert between oxidized and reduced. And in fact, a very common test we do in the microbiology lab is for a particular cytochrome. It's called cytochrome C. There's a huge class of organisms that don't have that cytochrome, and there's another big class that do. And so we can tell a difference, what we call an oxidase test, that actually we don't usually do in lab. I'm not sure why, but maybe because it's a little more expensive. I'm not sure, but that's just a little extra there. There are also carriers called ubiquinones, like coenzyme Q here. It's just a non-protein electron carrier. So again, you know, if I mention one of these, that should click, oh yeah, that's some kind of electron carrier in an electron transport chain. FAD drops off its electrons in a little different place than NAD. It's a little bit further down the hill. So we typically get a little bit less energy produced per FADH that drops off its electrons there. I would say ignore these yellow ATPs here because that's not really where the ATP is being produced. The next graphic shows the production of ATP a little bit better, but there's a big enzyme kind of here at the bottom where the protons fuse back into the cell, and that's where the ATP is produced. The reason that they put them in here is to just signify when about enough protons have been pumped outside the cell to equal 1 ATP. But I think it's more confusing there than anything. Okay, let's see. Stop with kind of questions, if you have questions. This picture shows some other things a little bit better in terms of, you know, where it's taking place in the cell. That's helpful. And it does a better job of making us aware of the proton gradient. Okay. So again, this represents all this potential energy in terms of protons outside of the cell, because the only place that they're allowed to fuse back through is at this enzyme complex called ATP synthase. Okay, so lots of potential energy out there. If you remember last time, I showed a picture of a dam. Okay, so when a dam holds back the lake and then lets it flow through in just one spot, that's, you know, can generate electricity from that. Same idea here. The protons outside of the cell, that's the lake, and then when it only diffuses in one spot, that powers the production of ATP. The other thing that this shows a little bit better is the final electron acceptor. So in this case, saying it's aerobic respiration, we have an oxygen accepting these electrons and producing water. So actually, it says half an oxygen. I think maybe what happens more often is you get 2 oxygens that combine with 4 electrons, and then you get 2 molecules of water. But this splitting of oxygen, that doesn't always happen quite correctly. The electrons don't always situate appropriately, and this is where we get toxic oxygen compounds. Like we talked about, you know, a week or two ago, like the superoxide dismutase, the peroxide anions, and actually a pretty high percentage. I can't remember what the percentage is that I read. It was something like 6, 7, 8% of, you know, the electrons that come through here end up making toxic oxygen compounds. So an organism that's has to be able to detoxify that with enzymes like superoxide dismutase, like catalase, like peroxidase. Okay, and sometimes we get some just leakage of electrons instead of getting passed by the carriers. The electrons might leak out. Remember, the electrons have to be chaperoned in the cell. The protons, they don't cause any damage. We can just kind of let them go outside the cell, but those electrons, again, can combine in inappropriate ways and cause damage and form those toxic oxygen products. Okay, let's see, what else did I want to mention here? Okay, so this proton gradient here, we often refer to that as a proton motive force. Okay, again, lots of potential energy outside of the cell there. Okay. And I gave myself a little extra to write there. So that's term, I wrote this out for you a little bit more clear. That proton motive force, again, that's all those protons that are building up outside of the cell. When they diffuse in down the concentration gradient, high concentration to low concentration, that phosphorylates ADP to ATP. So these two terms, proton motive force, and sometimes you see the term chemiosmosis. They're terms that are really closely related, but the proton motive force just really refers to, again, the potential energy, and then chemiosmosis, that's what's happening. Those ions are osmosing, if you will, through the cell membrane, which generates the ATP, okay, due to the establishment of that proton gradient. So there's kind of a few different ways to describe it. Those terms are really closely connected, kind of slight difference in meaning there. Okay. All right, so if we try to kind of add up the theoretical output of aerobic respiration, let's see, we talked about glycolysis and citric acid cycle there. So if we look at the output of ATP from electron transport chains, those 10 NADHs here and here convert into about 30 ATP. So we figure you get about 3 ATP per NADH, a little bit less, typically 2 ATP per FADH2, because it, again, comes into the electron transport chain a little bit lower down. Okay, so if you add all that up there, again, this I should mention, this is very theoretical because bacterial cells actually use a proton motive force to power flagella. That's different. Eukaryotes actually use ATP. Bacteria use proton motive force. So if they're using, when those protons diffuse in at the ATPase, that's actually not making ATP, it's just powering the movement of the cell. So if a bacteria is moving all around, you're not going to get this theoretical ATP yield. But at least in theory, for prokaryotes, if you add this all up, I think it's 38 ATP per glucose. In eukaryotes, typically we say a little bit less because they have to spend some ATP transporting the NADH into the mitochondria. Sorry, I think I'm getting sloppier as the hour goes on here. Okay. 38 ATP per glucose. Yeah, sorry. Okay, so again, that's pretty theoretical, but kind of that's what we like to figure out. Okay. All right, so the time we have left, I want to talk a little bit about, okay, so what if there's no glucose around for a cell? Can it use something else, or will it just die? Will it run out of energy? We're talking bacteria now, can catabolize proteins instead of glucose, or they can catabolize lipids instead of glucose. Again, it's not their first choice. If glucose is there, it's going to use that. But if they're going to use proteins to try to get some energy out of that, this is how they might do that. And this is a real simplification. This is the graphic from your book. I think there are other ways that this can happen, but I like this because it does tie into some things that we will be doing eventually in lab. Remember that bacterial cells, if they want to phagocytize a protein, they can't do that. Okay, they've got this cell wall. They can't extend any pseudopods. So if they want to break down a larger molecule, they have to secrete some kind of enzymes in order to do that. So a protease, again, this is going to be secreted outside of the cell. We might call that an extracellular enzyme or an exoenzyme. And remember, proteins are made of amino acids. So that protease is going to cleave those amino acids. Okay, and the amino acids then are small enough that they can be brought into the cell. Is that music somewhere? Okay, I try to yell at people for doing that, but okay. All right, so say it's then bringing an amino acid into the cell that can be transported in pretty easily. We have a deamination reaction. So that means the amino group is going to be removed from the amino acid. Next week in lab, if you're in lab, you're going to test for an enzyme phenylalanine deaminase. Okay, it removes that amino group from phenylalanine in order to get some energy out of it. Okay, so, and then you end up with this 2-carbon compound here that can be shunted into the Krebs cycle. And of course, then we get lots of electrons from that, a little bit of ATP by substrate level phosphorylation and energy at the electron transport chain. The other thing I wanted to point out, when you've got this NH2, that gets converted into ammonia. And typically, that's just a byproduct of the reaction. A cell might be able to use that as a building block somewhere. But what do you know about ammonia? What's the pH like of ammonia? Okay, it's very basic. Okay, so that's important because typically when an organism is catabolizing a sugar, almost all bacteria get really acidic end products. And happens more when they ferment sugars, but still to some extent when they go through cell respiration, it's always going to be acidic. So if the cell's using a protein instead, you're going to get very basic end products. And so if we have a pH indicator in the media, you know, we can kind of understand a little bit better what's happening there. Okay, so again, that's simplified, but kind of some important basics there about using proteins. A cell might also want to use a lipid for energy. And this happens in a step-by-step process. Get lots and lots of ATP, lots of energy from the catabolism of lipids. An enzyme like lipase, what is a lipid? It's actually made up of fatty acids and 3-carbon glycerol. Okay, and this is an enzyme that typically we test for in labs, so I always like to bring it up in lecture. That's not one of the things we're going to get to this semester. But that lipase does that first hydrolysis step. It separates the glycerol from the fatty acids. Okay, so that 3-carbon glycerol actually gets shunted, you know, spending a little ATP, and that gets changed into dihydroxy acetone phosphate, which that's one of the, what you get after you split, the splitting step in glycolysis. That's usually converted right into glyceraldehyde 3-phosphate. So that gets shunted right into glycolysis, and you know, you get some ATP from that, and pyruvic acid goes into the Krebs cycle, all that. What happens to the fatty acids here? They go through a process called beta oxidation. Okay, and again, this is a kind of a simplification that makes it hard to understand what's really going on here. But the beta carbon, that's the second carbon from the end, gets a carbonyl group added onto it. So that's a double bond oxygen. And then the two carbons are cleaved off. So in that process, you get some reducing power here, and you end up with an acetyl-CoA. Okay, and that happens again and again and again and again. All right, so all these acetyl-CoAs enter the Krebs cycle, lots of electrons from that, even more electrons here. So we get lots of ATP during beta oxidation. Okay, so recognize that beta oxidation, that's basically the process, catabolism of fatty acids. Again, we get, again, lots of energy from that. So that's the big picture there. Okay. All right. Let's go ahead and start the anaerobic respiration. So we're almost done with respiration. We're not going to get to fermentation today, but remember, I said anaerobic respiration just involves some other electron acceptor besides oxygen. What other electron acceptors can we have? We can have nitrate. And when that accepts electrons, it gets reduced to nitrite. And that's with an enzyme called nitrate reductase. Write that better. And that's another one of the enzymes that you're going to test for in lab next week. So I wanted to spend a little bit of time on that. This was not something that I posted, but that enzyme, nitrate reductase, it's right in there, right in the electron transport chain. So if organisms have that enzyme, they can use nitrate as a final electron acceptor. And again, still getting lots of energy from the electron transport chains. So it enables a huge number of bacteria to respire anaerobically, to be able to live in places where there's no oxygen. Okay, this is a huge family of bacteria. We usually abbreviate that and call it enterics, that you'll be dealing with in lab a little bit. But this is not something that we can do because, again, we don't have this enzyme, and so we can't use nitrate as a final electron acceptor. Okay, I think I'll pick up there next time and talk about, just briefly, a few other ions that can be used for anaerobic respiration. We'll talk about fermentation, and then we'll get into genetics. So I guess have a good weekend. Come see me if you want to look over your exam.